So we can start. My name is Martin Grötschel. I am the chair of the Einstein Foundation, and I have the pleasure to start this event tonight. I welcome you to this particular lecture, and the series is called Meeting Einstein. This is the ruin of the former Rudolf Virchow lecture hall, and I think it's a really unique location. It's very hard to find something similar elsewhere. This was the lecture hall of the famous pathological museum. It was destroyed towards the end of World War II by a bomb attack. Since the middle of the 1990s, this has been preserved as a ruin and is now used for some kind of formal, or let's say more informal events and for scientific exchange.
Thanks very much. So this is so I can pretend to speak German, even though I don't. This is what I'm going to talk about. If I knew more, I would tell you how the brain works, but I'm unable to do that. So instead, I'm going to try to convey a sense of why it's so difficult. So this is to summarize what it is that I want to say and give a sense of what I mean by understanding the brain, because people have different views of what that means. So first I listed some reasons why people might do neuroscience. This is not an exhaustive list. For example, I met a postdoc at the Salk Institute, and I asked him why he was studying neuroscience, and he said jobs are hard to find in physics. But that's not on here. So the three things that I listed were, one is to understand and be able to treat certain kinds of disease better, to be able to imitate the brain. This is why IBM hired me, in the hopeful but not necessarily sensible belief that one could do that. And finally, a more philosophical goal is to better understand who we are and what makes us human. Oh, okay, thanks. So now, how would one go about that, and how would we know if we understand something? So to me, what that means is to understand it from a physical point of view, which means being able to relate something that the brain does, which we can measure and quantify, and be able to account for it in terms of the physical properties of the neurons and how they interact with each other. And I'll give some examples where one can actually do that, and I'll try to give some examples where I think you can't do that, or we haven't been able to yet. So the sort of examples where you can do it are, for example, central pattern generators in invertebrates, and I'll show a picture. And there are certain kinds of activities in mammalian or in our own brains which we can account for it in this style, at least up to a point, but they're not thinking, they're rather sort of redundant. What does redundant mean? It means that the cells are doing pretty much the same thing. And you can characterize the population by looking at one or a few cells. So with that kind of thing in our own brains, we can give explanations of what's happening that look very reasonable. On the other hand, to account for thinking or cognition in this type of program has proven much more difficult. And to me, the main reason why it's been difficult is that we don't have a preparation where we can simultaneously study the output and study the circuitry. Oh, thank you. Schnapps, thanks. So just to set the stage, this is an actual conversation that took place in English, though, which is translated with the help of many friends. This is between me, the opa, and my grandson. So I'll just read it. Warum hat denn jeder Mensch ein Gehirn? And he said, damit wir denken können. Ja gut, aber wie ermöglicht das Gehirn uns zu denken? Kann ich noch einen Keks haben? So this child impressed me very much because he knew enough to be able to say he could not explain why the brain is able to think, and if only the whole field were as smart as he is. On the other hand, it also made me feel humble because if I knew how it worked, I would be able to explain it to him. And even if I could only explain it to him partly, I could explain it to his father, who's in his 30s, much better. But I can't do that. So let me give a sense of what one can do. So this is to convey some picture of what we're up against. Farid was my former student. This is a Golgi stain of a little bit of mouse cortex. These cells are all dead, so they're not actually doing anything. They're just sort of displaying themselves like models. But you do get a sense of how many there are, even though the stain only shows a small fraction of the cells that are really there. You get a sense of how big individual cells are. So one of these cells actually will extend processes, at least some of them, across most of the extent of the cortex. And then the output lines of these cells may go to the tail or near the tail of the animal. Or in the case of a whale, if you think of that, it's a long way from the head to the tail. So they're big. There are lots of them. They're complicated. You sort of get a picture. Now, to people or many people whose backgrounds are in computer science who want to make models of the brain, and I know such people at IBM, for example. A neuron is a neuron is a neuron. They're all equivalent. They all do the same thing. They're all sort of simple in shape, and in fact, that's not true at all. So this is an original drawing of Ramon y Cajal from the collection in Madrid. And it just gives a feeling for the diversity of shapes of cells. This is a Purkinje cell in the cerebellum, and these are granule cells. They come in all shapes and sizes and functions. Of course, you don't see the functions here. This is not sufficiently appreciated by people who don't actually look at the cells. This is another reason for the complexity and the difficulties we have. Now, I don't know where the idea came from that the mind is connected with the brain. If I knew more of the history of medicine, I would be able to say something. I can guess that it had something to do with two things. One is observations of the effects of a blow to the head, and the other was noticing that sense organs, like people knew that you hear with your ears and see with your eyes and taste with your tongue. These organs, or most of these organs, are actually in the head. So they're close to the brain, and maybe that was something that suggested that the brain was actually involved in them in some way. The notion of trying to localize functions within the brain goes back a very long way. This I picked because it's from Germany, and this is from the 16th century. So memory is here, and vermis is a worm. I don't know what that means here. And this is a sense of something. So this is an imaginative attempt to link parts of the brain with different functions. And we still do this with fMRI, and I guess we've progressed a little bit since this kind of picture. So this is how this view of the brain progressed over a few hundred years. I picked this also because it came from Berlin. So this is from Oskar and Cecile Vogt, and they were using data from monkey experiments, or mostly monkey experiments. They may also have had access to clinical observations from the Paul Broca school and other schools of so-called localizational neurology. You see a patient with a stroke or a tumor in some part of the head, and you look at what's gone wrong. But with monkey experiments, even in these days, this is in early 20th century, you could stimulate the cortex of the monkey and you might see what happens. So for example, the eyes might move to the opposite side of the stimulation. So they did this mapping, and it's hard for me to read. It's probably impossible for you to read. But there are these precise correspondences between different motor and sensory and eye movement and other sorts of functions, and then they transferred the monkey data onto a diagram of the human brain. So you can actually see that lots of territory here is left completely blank in this kind of experiment. It turns out these territories are just the ones which people who do cognitive neuroscience tend to record from these days. So you can ask yourself, suppose one could fill in this whole picture in this way. What would we have? What would we be able to do? So this is to give you, in a perverse way, a perspective. Suppose you could do that. How do you account for this? So this is an interview between a psychiatrist, the interviewer is a psychiatrist, and a patient who is diagnosed as having schizophrenia. So the psychiatrist says, have you been nervous or tense lately? No, I got a head of lettuce. You got a head of lettuce? I don't understand. Tell me about lettuce. Well, lettuce is a transformation of a dead cougar that suffered a relapse on the lion's toe. And he swallowed the lion and something happened. The Glory and Tommy, they're two heads and they're not whales, but they escaped with herds of vomit and things like that. Now you might think this is beat poetry from the 1960s, and I think one could write a computer program that would say there's something odd about this, sort of using some statistical methods. But it would be very hard to write a program that could distinguish this, which we recognize as being off, from something else, which we would say is poetry. And even if we could do that, you would have to say, how is it that information which is encoded in that form, how is it going to account for speech production at all? And any kind of content which there is in speech, how could we do that? And obviously that's a rhetorical question. So what kinds of things can we do? So this to me is the most beautiful sort of model of what one can do, along the lines of accounting for population behavior in terms of neurons and their connections. So I downloaded this picture from the web, and I'm sorry, I don't know who originated it, but let me just say a little bit about what it is. So a lobster has its teeth inside its stomach. So there are 3 teeth, and when the lobster eats something, the stomach can contort itself in different ways. And this has actually been studied with endoscopy. So some of you here may have had endoscopy if you have an ulcer, say, of your stomach. But some did, in German actually, did endoscopy in the lobster stomach. And with different stimulation or neuromodulators, so-called, or with contents in the stomach, there are different modes of action of the stomach and grinding of the teeth. So it's a bit like a washing machine. So a washing machine has a small number of modes in which it operates. You know, water can come in, water can go out. It agitates clothes, it spins. And this is analogous to that. And your washing machines will have a little chip that controls the modes, and you can set some parameters like rinse this long or spin this long. And in the same way, you have a chip or a set of cells of order 10 or 15, and they've all been mapped. So if you have 2 individuals of the same species, this circuit, this so-called stomatogastric ganglion circuit, each cell is identifiable and can be given a name, and they're the same in different individuals of the species. And then it's possible to map every single connection between every cell. So these resistors here correspond to what are called gap junctions, and these other lines are synaptic connections, and they can all be characterized by recording from pairs of cells. And then in addition, you can isolate individual cells and study what they do in response to an artificial stimulus, and say something about the so-called intrinsic properties of the neurons. Now you can do other things too, because this generates a motor output. So when individual cells are active, it's possible to draw a correspondence between what that cell does and what the animal is doing, or in particular what tooth is doing what, or which part of the stomach is doing what. And that means that you can actually remove the whole ganglion from the animal and study it in isolation. So it's generating, it's called fictive locomotion. So nothing is actually moving when the preparation is in vitro, but you know what would move if the preparation were doing this, that, and the other. So because you have a small number of cells and you know something of their intrinsic properties and the connections, it's been possible over the years, and Eve Marder is involved in this, and Alan Silverstein and many other people, to develop very sophisticated models that account for how the circuit operates under different conditions of stimulation, of laser ablation of single cells, of putting chemical compounds in the bath, and you can account for it. Now, we don't want to get too carried away, because these cells here are shown as circles. This is what they actually look like. And when an experimenter puts an electrode in one of the cells in the bath, they're actually recording from there. They're not recording from here or from here. And in fact, almost nothing is known about what's going on in these other parts of the cells. So this is an idealization, but it's as nice as an idealization as one can get. Identifiable cells, defined circuitry, defined output. So can we take this paradigm and continue it to more complex sort of circuits? And what happens as we do this? So I just picked a few examples. So this is a central pattern generating circuit in a vertebrate. So it's a weakly electric fish. So these fish live at the bottom of the Amazon and Orinoco rivers in South America in water that tends to be turbid. And vision doesn't work very well. So what the fish do is they take modified muscle cells and use them to produce electric fields, and so currents flow in the water around them, and they can use these to sense what's happening around them, and also as a way of communicating with other fish. And they actually sense each other's fields and adjust the frequencies of their own fields so that the frequencies will repel one another. These aren't the fish that kill or shock animals with very large fields. These are weakly electric fish. So they're interesting. This circuit is interesting because the electric field oscillates at 500 hertz or a kilohertz or even faster. And there are highly specialized cells in the medulla of the fish which are connected together, which generate this output. So it's more complicated than the stomatogastric ganglion. There are more cells, approximately 200 cells. You can't map each one. There are these big ones and then the little ones. So what you end up with is, you know the number of cells, you know what they look like. You can study this preparation in vitro and do experiments, but it's not a situation with identifiable neurons. You can also measure the output of the system in vitro, and in that sense it does have something in common with the stomatogastric ganglion. So then what is another level of complexity? So I picked this example. So Sten Grillner in Stockholm has been studying this preparation for many decades, and what it is this sort of disgusting kind of fish called a lamprey. This is a jawless fish. So the mouth of the lamprey doesn't do that, but it's actually like a hole, and it has hooks, and they attach themselves to other fish and parasitize them. But what primitive fish and reptiles, one of their properties is that you can do considerable violence to them and take out parts of their nervous system and study big circuits in vitro. So even with mammals, like many people here, can take a slice of mammalian tissue and keep it alive, but that's just a slice. Here you can remove the whole spinal cord of the animal, and you can put it in a bath and record from it and stimulate it. and you can observe its output. So again, it's like, because it's a motor system and because it makes the animal swim, each of the roots, the nerve roots that come from the spinal cord, generate signals which have meaning to the experimenter because they have meaning to the fish. So you can record activity in a nerve root, and you know that in real life this goes to certain muscles, and when that root is active, those muscles will contract. So even though you have a spinal cord that's sitting immobile in a dish and not moving at all, in fact, you can make it swim. It's a sort of artificial swimming, but it's defined. So you can sort of see where I'm going. You can't make it think, or if you did make it think, you wouldn't know that it was thinking because you don't know how to measure thinking. But you do know how to measure signals that would go to muscles and make them contract. So what can you do with this? Well, when a lamprey swims, it can swim forward or backwards. It can swim at different speeds. So this is, of course, swimming when it's not attached to a host. This is sort of swimming alone. And the swimming has the property that whatever speed it's swimming, there's one cycle along the lamprey. So that if you look at this picture, you start here and you go here and you come back to here. So the lamprey swims like this, but at any speed, it has one cycle. So as opposed to doing this. So this led to some mathematicians like Nancy Koppel in the 1980s asking, how can you do that? How can you have a distributed set of neurons connect together, generate an oscillation which has these properties? And in addition, the speed of the oscillation is set by inputs that come from the brainstem, somewhat in this region, and which mostly go just to one end of the spinal cord. So the whole system is under control of a localized piece of brain, and it makes these oscillations with these properties. And what this picture shows is the phase relations between different segments of the spinal cord during a swim. So the Stockholm group has actually done enough recording, has characterized enough of the anatomy of the cells and the properties of the cells to come up with a very detailed model that actually accounts for these observations. Their model has, of order, I forget, tens of thousands of neurons. They're excitatory, they're inhibitory. This is the E and the I here. These are the two sides of the spinal cord, and you can sort of see the phase relations. You can do experiments in which you cut the cord this way, or you can cut it longitudinally, and you can explain what happens. This is very beautiful. Okay, so again, what about mammals? So let me back off a minute. Now, suppose we want to continue this sort of program with mammalian tissue. What can we do? How would we go about it? So here are some of the issues that we face. As I said before, neurons, mammalian neurons, at least some of them, are very large. Like this pyramidal cell has this enormously long dendrite. So this is cortical layer 5 here. Dendrite goes up to layer 1. This doesn't even show the axon. Now, another problem we have is that different parts of these neurons do different things. So this is from the Sackmann school. If you do a recording here out in the dendrites, or if you do a recording here at the soma, or if you were to record from the axon, like some of the people in the audience here do, almost as I speak, you see very different things. So in this dendrite, there's this action potential which is extremely broad. This is 10 milliseconds. And if you look at action potentials on the soma, they look like this. They're very narrow, more like 1 millisecond. And the interaction between slower events here and faster events here makes these complicated intrinsic patterns of activity, which I can't go into in detail. The point of it is that this is not it's not a uniform structure, and it's absolutely silly to think of this as something which just generates spike trains. So a better sense would be that this is the cell has a sort of a mind of its own, and and even each little piece of it has a mind of its own. This is something that I learned from Rodolfo Linas in the 1970s, and it's more true now than it was then. So that the intrinsic properties of our neurons really matter for what's happening. Okay, but does that mean we can't do anything? Well, no. So I want to show some examples where we can make very detailed explanations of experimental data that involve populations of cells. So this is physiology in a single cell. This is population activity in 1,000 cells. In the case of the model, it's 100 cells, but in the preparation, it would be a few thousand cells. So let me try to explain what the preparation actually is. So this comes from a mammal. It actually came from a guinea pig. A part of the brain called the hippocampus was removed from the animal. The rest of the animal died, but this part of the hippocampus was kept alive and put into a bath of oxygenated and glucose-laden solution, which kept it alive, more or less. It wasn't in a normal state. Many of the parts of the cells have been shaved off in this procedure, but the cells were alive, and they also were connected together, at least partly. And they were connected together enough so that if you perturb the system, so this is adding a drug which blocks some of the connections between the cells, you get this phenomenon like this. This is an average signal. It goes brrrr for about 50 milliseconds. And what's happening is every single cell is making a burst of spikes. So here, in this previous slide, this is a burst of spikes in a single cell which has been evoked by a stimulus. And here you have many cells, and they all burst together. Except a few of them do this, they burst initially. So it's possible to account for this kind of behavior with a model, and the model has good features and it has bad features. So let me just say how this is different from a central pattern generator. The central pattern generator, each cell has its own identity and its own function. In this system, none of the cells have an identity. They're not identifiable. They're not the same from individual to individual. It's impossible to make a map of the connections. What we can do instead is say something about the statistical properties of the connections. So we can say, on average, each cell connects to so many others, and more than one other. So it's above what's called the percolation limit. So that's a statistical statement of about the connectivity. We can't characterize the intrinsic properties of each neuron, but we can say most of the neurons, if we were to isolate them, would do something. And we assume that in the preparation under these conditions, they do the same thing. Now, we can also predict that at a connection from one cell to another cell, something happens. We can't say exactly what happens, like we can in a lobster, but we can say, on average, what happens in the transaction from going to this cell to this cell, something happens. And you can put that together and you can describe what I just said in precise terms to a computer. The computer can do its thing, and it generates wiggly lines, and the wiggly lines look like the experiments. So this sounds great, like, oh, well, we've accounted for this behavior. Well, we have, and we haven't. This is a behavior which is, a physicist would say, is highly degenerate. It's characterized by the behavior of a few cells, even though thousands of cells are actually generating it. That's one thing. Another thing is this activity is going on autonomously. So here's a piece of tissue, it's sitting in a bath, it sees no sensory input, it's not generating any output that's recognizable to the animal or that's recognizable to the experiment. So it's an autonomous system, just doing its thing like a physical preparation, and we can make a model of it. So we have something, but we don't have what we think we have. We don't have what we'd like to have, which is something truly analogous to the central pattern generator. So now I want to touch on something that Dietmar alluded to. So this has to do with what we were working on when we met 15 years ago. And I'm allowed to be more provocative than he is because he's an experimentalist and I'm not. Yeah, so I can play fast and loose in a way I don't think Dietmar would like to do. So what I will say is that there's a dogma in neuroscience that here's how neurons work. They have lots of synaptic inputs and they integrate their inputs and they generate outputs. And this has sort of been the basis of most models of the nervous system since the 1930s, like McCulloch-Pitts neurons, that's early 1940s. It's collect inputs, integrate, generate outputs. But in fact, that's not always true, that neurons have other ways of generating outputs which are completely independent of their synaptic inputs. And this is an experiment which shows that this is a behavior of one cell of many, and the whole population is doing this kind of thing and generating an oscillation without synapses. So this is purely so-called electrical or gap junction coupling between the cells. And not only that, the activities that you see when you record from the soma are not all action potentials. There are different kinds of action potentials that correspond to where the spikes are generated and or how they propagate in the cell. So this is not totally out of line with what I've shown before. So these cells have connections by gap junctions, all these resistors. Many of the connections are through gap junctions plus synapses, like with us. The difference is that in mammalian brain, the significance of gap junctions tends to be downplayed, and this is a mistake. Now, so where can you go with this style of modeling and explanation of nervous activity? Well, one direction you can go is in trying to understand epilepsy. So this is the kind of signal you would record from the surface of the brain of a patient. This is a small child who is having a seizure. This is the seizure here, this very high amplitude rhythmical activity. And each one of these events corresponds sort of, to one of these. Not exactly, but approximately to this. So we have some understanding of that. And each of the seizures in this child and in many other patients begins with this high frequency network oscillation which corresponds at least approximately to this. So even though we're studying networks of cells which are characterized statistically and where the output is not cognition but just an oscillation or bursting or something of that sort, it's still useful. I don't want to dismiss it. It's useful, but it's not addressing this most basic question of thought, and we need to keep that in mind. Now, can we go further? Well, a bit. So here's another kind of activity, also in a slice. So a piece of brain which is disconnected from the rest of the brain and hence from the rest of the universe. It's bathed in a drug which is sort of a poison and which unmasks an autonomous oscillation. And this is time frequency. This is time here in seconds. This is going from 0 to 20 seconds, and this is frequency. And what this is showing here is there's this run of oscillations at 40 hertz. So here in Germany, I mean, this 40 hertz rhythm was discovered in Germany in two places. So there's in Frankfurt with the Gray and Wolfsinger, and I guess in Marburg with Reinhard Eckhorn. And they were looking at a 40 hertz oscillation which is evoked by sensory stimulation. And that kind, and there's been much discussion here about that, but that kind of evoked 40 hertz is different than this. That 40 hertz is transient. And this kind of 40 hertz, which is produced by a drug, is not transient. It just persists for hours. But, so this is in superficial layers of the cortex or the cortical slice. There's another oscillation which is produced in the deep layers, which is called a beta oscillation, that goes at 25 hertz. So let me just say a little bit about that. Again, it has the same property that one can give a statistical account of the connectivity and the behavior of the cells, but not a detailed account. Detailed meaning the activity of each cell carries significance and can be described in a code or a language. So this is the kind of thing you see in the experiment. This is an average signal, and it's just buzzing along at 25 hertz, and this is what a single cell is doing. It's doing something like what I showed before. It's doing something like that. But it's coherent. So even though this cell doesn't make a full spike on every wave, the population is producing an oscillation. And this is the model. Now I picked this because this oscillation is produced without synapses. So this phenomenon is entirely mediated by gap junctions. And even though this is a slice, and I don't want to say this is a completely normal kind of activity, it looks like activities that are normal. Namely, during preparation for movement, you can record oscillations at this frequency in the same parts of the brain where this is produced in vitro. So this kind of thing might be going on in our brains normally. It's very hard to prove that, but it might be. Now, does this tell us how we move or how we decide to move? Absolutely not. It does not do that. What it does is it tells us something about what might be a substrate which allows us to generate particular movements and not other movements. So when I was at IBM in the 1980s, I had this conversation with a solid-state physicist, of which there are many at IBM, or more in those days than now, that we're getting out of hardware. And anyway, he said, "What are you doing, wasting all your time with these details? We have a model of the brain." So that was a surprise to me, and I asked what he was talking about, and he said, "We have the Hopfield model." So, okay. So let me just say a little bit about what that is. So this has other names, depending on who you talk to, who says he or she invented it. But this is a commonly used name. And this model came out of physics. But the qualitative idea is this: we're never going to know all about neurons. We're never going to know how they're connected. We're never going to be able to study the behavior of a big piece of brain and really understand it. So what we should do instead is say, well, there's some qualitative features which must emerge, however the brain is connected and however the neurons work. There are certain qualitative features which have to emerge out of that. And so what would one of those features be? Well, there's something called an attractor. And what that would be is, if you think of motion of a particle in what's called a phase space, and it might be wandering around, but it would want to settle down, maybe spiral around to a fixed point, and that fixed point would be an attractor. Or there might be an orbit like this, and the thing might wander around, but eventually it gets closer and closer, and it just approximates this orbit forever. And then you could call the orbit an attractor. So the insight was that, well, a memory, if we have a memory of something, it acts like an attractor. And what the neurons are doing when we remember is we have some idea what the memory is, and then they converge onto this so-called attractor. And then, so let's make a mathematical model of that. And so how will we do that? So this came out of ideas of something called a spin glass, or also of magnetic materials, So I'll try to explain it. If you have, say you have a magnetic material, has lots of atoms, each of them has a spin, and the spin can be up or down. And suppose you have two atoms, and you can write down how they talk to each other, or how they influence each other. So it might be that if this spin is up, it tends to increase the probability that this spin is up. And you can describe that exactly. So now it turns out that in many of these systems, they exhibit a set of so-called attractors, which means there's some domain in which all the spins are up in this domain. And if you get close to it, you converge to it. So it acts like a memory. And in this model, these are the actual equations, but I won't go through them. In this model, it shows how you can actually exhibit particular attractors and examine their properties. It's very beautiful, actually, as mathematics. Now, the problem with this as neuroscience is it's been impossible to prove. So the late Danny Amit, who is an Israeli physicist, was very interested in this, and he actually wrote a book arguing that data from monkey experiments showed that this model had been confirmed. So you can read his book and make up your own mind about it. From my point of view, this has not been confirmed. There is no actual preparation in which you can observe these quantities and show that an attractor exists. It's not to say they don't exist, it's just been impossible to demonstrate. So my take on this, or my spin on this, no pun intended, is that there may be shortcuts, but we don't know if the shortcuts are right. So these are the real cells. This is not a spin. I mean, maybe my talk has a spin on it, but that's another question. So, so let me just repeat what I've been trying to say. We want to treat disease. I showed two examples of disease. One was schizophrenia, and I don't think this talk says anything about schizophrenia. People can argue about that. But maybe it says something about epilepsy. To learn how to imitate the brain. This is something, of course, IBM would like to do, and is extremely difficult. And there's controversy, of course, whether you need to know how the brain works in order to imitate its functions. And then finally, to understand our cells. I showed you examples of where you can actually relate physical properties of cells and connections to the output, although I didn't go into any real detail. And that was with the central pattern generators and with seizures and network oscillations. But with cognition, where's the hang-up? The hang-up is we can't define cognition in such a way that we can measure it in cellular terms and relate it to the properties of cells and networks. We don't know how to do that. And for that and for other reasons, we don't have a preparation where at the same time we can observe an output which makes sense in cognitive terms and also makes sense in physiological terms. I don't want to say that it's impossible, it's just that from my point of view, no one has figured out how to do that. To give an example of each of these goals. So I'm going to try to do that. These are, of course, selected examples. So here is neuroscience not answering a question, but posing a question. So many people study this phenomenon called sharp wave ripples, which occur normally in the hippocampus in rodents. And during one of these events, there's a transient and an oscillation at, say, 200 hertz. And this is the average signal, and this is a cell. And this spike here is very strange. This is not how cells are supposed to spike. Cells spike in classical dogma because they get excited by their inputs, which means they go like this. This cell is being inhibited, but it spikes anyway. Now, this signal here, this is from resected human tissue. So this was from a patient who had epilepsy, who was being worked up for surgery to remove part of the cortex, had in situ recording, had the surgery. Some of the tissue removed at surgery was kept alive and studied in a chamber somewhat, not exactly, somewhat like the chamber used to study this tissue. And what happens is very similar. So this signal here is filtered, so you're not seeing the wave here, but you're seeing a high frequency oscillation which looks very much like this. And the spikes look a little bit different, but they have a similar property. They suggest that these spikes are coming from the wrong place or from an unusual place in the cell. Now, this is clearly pathological. This is normal, and not only normal, may be important for memory consolidation. So here's the question. If we want to treat epilepsy with medicines rather than with surgery, it might be nice to try to abolish this kind of thing. But wouldn't we also abolish this? So it means this is a scientific question. What distinguishes this from this? So this is an example of neuroscience at least pointing the way toward an approach to therapy. Now with technology, I don't know how to draw inferences from the brain that are useful in artificial intelligence. It's not to say people can't do it, it's just I don't understand how to do that. And what I hear is, we just have to copy the brain's algorithms and emulate them in software, which would be nice if we knew what the algorithms were. But we don't, and we don't know how, at least in my point of view, we don't know how to get at them. So here's another example. There's a field called brain-computer interface, or BCI, and the idea is to measure signals emanating from the brain somehow or other. Sometimes people do this invasively, or they try to do it non-invasively, and maybe those signals could be used with a patient who's paraplegic, or blind, or has some impairment that you could correct if you had access to the signals and could introduce signals into the brain and bypass the usual pathways. So it turns out, so people try to do this with EEG electrodes, and the bandwidth, the sort of bits per second that they can extract from brain activity is very, very small. And you can't really do a lot with it. Another approach which has been suggested is to use magnetic fields. So it turns out that because of the way the dendrites of cells are lined up, if there's coherent activity in lots of cells, they produce magnetic fields, which can be measured outside the head. They're very small, but they're detectable. And the way this is done nowadays is like this. So this child is sitting in a chair in a magnetically shielded room, which is an expensive operation. And what this enormous object is a huge thermos bottle. It's a dewar, and it's filled with liquid helium. And why is it filled with liquid helium? Because the magnetic detectors they're called squids. It's superconducting quantum interference devices, which is what you need to detect these very small fields. They operate at extremely low temperatures. So you have a huge amount of liquid helium in this object, so you can detect the fields that are being produced by his brain while he holds still. Now, this has no future in brain-computer interfaces, but technology is moving on, and there's a company and there's a professor, Yoshi Okada at Harvard, and they've developed a portable MEG device which can be used in babies, and because babies have very thin skulls. And they hope to be able to actually use this in a neonatal nursery. I assume the cryogenics is all down in here, and there's a pipe that runs from that up to this helmet on the baby's, or I guess this is a doll, but on the baby's head. And then there's a lot of software to get rid of all the ambient magnetic signals. So in any case, why do I show this? Well, if we're going to make sense of these measurements, suppose it's possible to do this with adults without having to have all this to do here. How do we make sense out of the signals? We have to understand what's going on inside the head. So this is a technological direction. And the last example I want to show is a question. So I downloaded this image this is a Golgi stained human pyramidal cell, and it's, again, I'm sorry, I don't know who actually prepared the cell, but that's a human cell. Oops. This is a rodent cell. They look the same. Whatever it is that makes us human is not in our neurons. This is something that is actually well known and not original to me. I just want to point it out, that whatever it is that makes us what we are, it's not the cells. I mean, it's true there are a few cells that seem to be peculiar to humans, but it's a very small number. It's something much more subtle and much more elusive about the design, and I wouldn't begin to speculate what it could be. And then finally, this is again to try to pretend that I speak German. Wovon man nicht sprechen kann, this has become a cliche in the US, I don't know about here, darüber muss man schweigen. And I want to thank especially the Einstein Foundation, of course, for the privilege of being here, but these individuals, Miles Whittington, who's in the UK, Dietmar, who is here, Andreas Dragun in Heidelberg, and Hanna Monnier in Heidelberg. And thanks very much.